Is that okay to have one speaker? Okay. So then I invite Dr. Odorisio, who is the next speaker on this session. You know a lot about Dr. Odorisio, so I don't need to say, but that he has very interesting new research studies that shows how DNA repair factors play a role in the resistance to chemotherapy. I'm quite excited that this result has not been published. Please note that. It is all fresh, hot from his lab. Tom, you have 15 minutes?
It's right here.
This is 2.4.
Okay, good.
If you don't mind, I'll give you a 5-minute and 1 minute.
That's fine.
You have 15 minutes to
Okay. Thank you very much, Dr. Shah. I appreciate the invitation once again to the Canadian Carcinoid Society group. Usually when I prepare for this, as I did 3 years ago with the first meeting, I think Toronto, I took out the list of faculty members just to get myself wound up a little bit and telling myself I could keep up with the fast dogs. And I read over it and I didn't sleep very well. And then I got up and started thinking some more about it, and I decided that just like that movie you probably all remember, some of you do anyway, Irma LaDouche, Jack Lemmon was talking to Shirley MacLaine, and he made a comment. He said that he was interacted in a battle and he was severely wounded, and then he said something to the effect that I'm just half a man, and then he asked Irma, he said, please be gentle with me. So I want you to know I'm not going to tell you what part of a man I feel I am, but I'm asking the group to please be gentle with me on this. This is a new talk and it's always a little risky, but I'd like to share with you, and I really appreciate the comments of Dr. Van Ruder already in the great introduction. My goal today is to walk us through rather quickly, and we have 15 minutes, and all of the important information I hope to convey to somehow explain the chemoresistance of these neuroendocrine tumors is in the last 6 or 7 slides. But to show you, as we've learned, that the white and the black are very easy to determine. The slow-growing ones are usually with a Ki-67 of less than 5%, and greater than 20%, they're much more aggressive. But it's that group 2. It's the intermediate group that we're all wanting to know what we can do and why they are the way they are. And I'm not saying we have all the answers, but I think some very recent information makes it very exciting. This is a cell I always show, and it has the locks on the cell. This is a classic cell of a neuroendocrine cell with a rich apparatus. The stain on that is a neuron-specific enolase, but you heard from Dr. Asa, chromogranin A and synaptophysin help define the essence of the neuroendocrine tumors. Whether they're the type 1 or the type 2, we'll see in a minute, of Solcia and colleagues, the WHO classification, or even the small cells. The neuroendocrine component of those cells are defined by the stains. The typing, we've heard multiple times today from the World Health Organization. It has changed a little bit in terms of further, more sophisticated staging, but I think Solcia and the group got it right in 2000. The two types that most of the folks that I see with midgut and the pancreatic neuroendocrine tumors are the type 1 and type 2. And you'll notice both of them start out with well differentiated. One is a tumor, and the carcinoma defines a little bit of invasion. But we'll see in a few moments that those two groups are put together when you look at a Kaplan-Meier curve of those two groups against the group 3 that are fortunately not as common, at least initially. And that is the poorly differentiated endocrine small cell carcinoma, which has very little staining granules of chromogranin A and a high mitotic index. And those are not the group that we're concerned about today, but rather type 1 and type 2. The well-differentiated, I'll just go right to them. The type 1 is the salt and pepper kind of pattern that we've heard about earlier today, very homogeneous appearing with clear areas around the nuclei. And there's a stain of that very same tumor intensely with chromogranin A. The well-differentiated neuroendocrine carcinoma, the so-called atypical carcinoid or the malignant carcinoid, is the ones that have nuclear pleomorphism in it. And then the poorly differentiated are the small or large cell carcinomas that you can see where they have large, bizarre-looking cells. This is an H&E. That brings to us the Ki-67, which we've heard a lot about today. It's an antibody that recognizes an antigen. It's encoded by a single gene in chromosome 10. Its expression is tightly associated with cell cycle, and it's an excellent indicator of tumor proliferation. MIB1 is a slightly different antibody from the same gene area, and it's also monoclonal and it's widely available. It is an antibody, and it recognizes the different epitope the MIB1 does. But this is a picture of a very high, increased proliferation using the Ki-67, and these are the ones that are usually greater than 20 or 30%. Again, the nuclear staining is seen with the dark dots. Now, a very important paper by Fagiano came out, and using the World Health Organization classification, they had 31 patients. The most important thing about this study is that they took the two groups together, the well-differentiated tumor and the carcinoma, against the poorly differentiated group. And they did a Ki-67, chromogranin A, and synaptophysin. And that was their scoring grading system. And what you can see there is the two groups. The top line, the long-lived line, have both types, well-differentiated tumor and well-differentiated carcinoma. So those are the two groups that do well. But within that group is the group we're all talking about, mostly affected, and how we can impact and why the chemo resistance is what it is. No one sees, we can all see why the high proliferation index group did not do well. So in summary, the Ki-67 or the MIB1 is helpful in pointing out prognosis and therapeutic direction. We still need to have that, and certainly the ENETS, the European group, has grasped on that and has insisted that the Ki-67. I will say this, that the Ki-67 and how it's done matters. The rules are 1,000 cells, 4 slides, carefully counted, period. And if you do it that way, you get excellent outcomes and consistent outcomes with the technique of counting Ki-67, as they do in most of Europe. I'm not certain the United States has adapted that careful study process to determine an actual proliferation index. Furthermore, we heard a little bit about this from Dr. Ardil. Think about cutting the salami. It's a piece of tumor. It's heterogeneous, not homogeneous, as we've heard today. Different distribution of tissue in the middle and at the end. So if you cut one end, your Ki-67 will look maybe one way. You cut the other end, if you have multiple blocks or a block of tissue being sent around the country or to different pathology groups to read, and the interpretation may be different. Furthermore, there's striking variability of the peroxidase staining from lab to lab. So you must needs be careful. When you all come and say, I need to know my Ki-67, you need to also get a grip. Because in a way, you don't want to put yourself in a corner and saying, my Ki-60, it's like, how are things going? Fine, my Ki-67 is this and this. But you have to understand it's very relative, as you've seen. The enormous range from less than 2%, which is most of the folks, to greater than 20% is not usually missed. But there is variability in between. This is a slide. I give credit to Dr. Barry DeYoung, our pathologist. Sue and I raised an antibody, the SST2, that you've heard about. And this is a slide of a liver tumor. And there's normal liver on your left. And this is now getting to the point of what we heard beautifully from Dr. Van Wouter, and that is the peptide has to lock into its membrane receptor, and then it's internalized. And this is the SST2 antibody. Dr. DeYoung, when he did the staining, saw this, and he said, well, this is a very intense stained tumor. There's the receptor stain of the tumor, and you can see membrane receptors staining, or membrane staining, that is, inside on the surface of the cell, and you can also see the cytoplasm inside the cell is also staining. and he actually broke it down into a predominant membrane pattern and a cytoplasmic pattern long before anybody thought about the grading system of these. And now we know why. This is another stain from Dr. DeYoung showing the cytoplasmic staining. That whole slide is loaded with just the complex. So octreotide or somatostatin has bound to the membrane and has gone inside the cell. This, you can all see the membrane around, the dark brown. That's an intense membrane staining, and that means this cell has a predominant membrane staining. Now, this was all very interesting, and this is one that's a whopping membrane and cytoplasmic staining. And you say, gosh, where is all of this going? We know from what Dr. Van Ruder said and what the literature says, that if it sits on the membrane, on the outside on the cell, it isn't doing very much. It's got to get inside the cell to do the mechanisms that it does. Anti-secretory, anti-proliferation, perhaps apoptosis, etc., antagonizing IGF-1. This is a premier paper, came out 3 weeks ago by Dr. Jean-Claude Royby. And what he did was what he does so well. He looked at these internalization of the membranes. He looked at it in real-time pathology. And what he showed using different antibodies on the top is the same membrane pattern. But below that, in the middle and in the lower panels, are the cytoplasmic staining. With exquisite techniques, he identified the same thing that Barry DeYoung did. But he did more than that. He traced the complex and showed that the somatostatin, or in this case sandostatin, bound to the membrane and went into the cell, and showed very clearly for the first time that that cytoplasmic staining is the complex. It's the whole ball of wax going into the cell. Very dramatic. But what he also showed is when the person wasn't taking octreotide, it was all on the membrane. When they took octreotide before surgery, it went into the cell. So somehow octreotide is driving the mechanism to take it in. This was a little paper as we get to the mechanism of why there is chemoresistance. This is a paper that an abstract paper soon by Juweed and Serbu in our group. And in this group, we knew about the DNA repair proteins, and in this case, we wanted to see if there were DNA repair proteins in neuroendocrine tumors. And lo and behold, we took samples from 17 patients with confirmed carcinoid and pancreatic neuroendocrine tumors of various WIC grades. And what you see on the left panel is the H&E staining, and on the right side is the Ki6, or the B panel is Ki67 in the lower left panel, and on the right panel is the PCA staining, a stain recognized for DNA repair, recognizing that it has two faces. Clearly it's a DNA repair gene. That means these cells in these tumors repair themselves, or are prone to repair themselves very quickly. In conclusion, we thought this was the first demonstration of a high expression of PCNA, and these were in 90% of the 17 samples that we looked at. So this is a very loaded. What separates it a little bit is the percent. They're going to have to be quantitated. It's not enough to say positive. You have to quantitate the percent. Finally, this is a stain of an ERCC. This is after the work in New England Journal of Medicine where they looked at this. This is it's like a pair of scissors, and it goes along and it clips out a mutated piece of a cell, and it repairs it. So now you have the question, is this protein, is this enzyme present in these tumors? We know about lung cancers, and in fact, half of them had, in the work in the New England Journal, half of them stained with ERCC1, the other half did not. Those that stained with ERCC1 did not respond well to chemotherapy. Those that responded with lung cancer that were ERCC1 negative, they responded very well to chemotherapy. So this is yet another repair factor in these cells. In the case of the first 10 we've looked at, all of them had 100% staining of the ERCC1. suggesting that you've got little factories in these cells that repair themselves. Are we surprised? If you go back and you look at what cell model, what cell neuroendocrine tumor have they ever been able to perpetuate and grow, there are darn few. These cells are resistant to being able to turn over very fast. They're very slow. They're a very stable cell. The Bond cell line has been clearly the one most used in vitro work and in transfer of tumors to animals. This is the reason that these cells are resistant to chemotherapy. It's because they've got little repair shops inside of them, and these cells tell the story. The original tumor tells the story, where the answer will be between type 1 tumor, well-differentiated tumor, and type 2 tumor, the intermediate grade. That gray zone between 5 and 20% will be in the percent of how much of this enzyme, this clipper, and the PCNA are there in those tumors. And we will have a very good handle on how to, on which tumors may respond best. Arthur alluded to Fagiano's paper and also alluded that the group that were 20 or 30% responded well to mTOR inhibitors. I haven't seen that, but that would certainly maybe make that a viable chemo or drug for it. Finally, we all have to say yes, but. This is the white and this is the black. On the left side is an Octria scan of the patient. 3 days later is the PET scan of the same patient, and both of them light up. All the tumors, or several of the tumors, some similar, light up in the same direction. So this patient was very strongly avid for Octria scan, positive receptors for SST2, and also had an SUV in the 7 and 8 range on most of the tumors. Patient was treated aggressively with cisplatin and carboplatin and VP16 for 6 cycles and put on octreotide acetate, had lost 35 pounds, is now 4 years on just octreotide acetate and doing very, very well. There's the white and there's the black, and this is kind of what we need to look at. We have to use tools when we don't have the accessibility yet of these kind of subtle predictors. But we can see that there is a way that we can still attack the glands reasonably with chemotherapy, and that is with a rationale. I still think the PET, FDG PET, the sugar PET as it was called, is still very helpful at the right time. I don't know exactly that time, but it seems to me that with their behavior by CT more aggressive, it should be done and it should be addressed. Thank you very, very much.